Here in the Guiltinan lab, we have a tissue culture pipeline and focus specifically on the Obroma cacao. The reason why the tissue culture pipeline was developed is because, principally, sometimes cacao is not self-compatible, meaning you can't breed the tree with itself. Also, between different varieties, sometimes they're not compatible. So if you have a tree that exhibits some really good phenotypes, like resistance to disease or larger pod size, you want to propagate that specific genotype. And the way that you'd have to do it is through tissue culture. We don't have any flowers here at the moment in the lab, but when we start, we always start from a flower. And we cut the flower into pieces, and the petals that are inside, we cut them and then place them onto a specific type of media. And what it usually looks like is something like this, where we would just lay out the 4 different petals from inside the flower, and they will start to grow. After 2 weeks, we transfer them to a different type of media, the secondary callus growth. And then after that, 2 more weeks, we transfer to an embryo development media. So this is at the very beginning, maybe about 6 weeks into the tissue culture. This culture was started in August of last year. Sorry, August of 2014. And we can see some of the different types or the development of the plant. From the callus, it'll start to have different embryos that develop, similar to this one here. It's just small and it begins to elongate the axial part, and then the cotyledons at the very end. Depending on the different types of hormones that are in the media, it starts to go through its process of developing embryos, and then the embryo develops without pollination into these small little plants. And then these will mature, and if you want to do cuttings from these, you can, just taking the cotyledons and starting the whole process over again. But this is a way that you can have flowers from one plant, and they begin. You can very easily have hundreds of plants in under a year. These are clones of the original plant. This isn't always necessary for every variety of cacao, but as I just mentioned before, some varieties cannot self-pollinate, and so this is the only way that you could propagate a clone or something that has those characteristics that you wish. Yeah. Usually the best chance that you have to propagate them is from using the petals of the flower. When they get bigger, you can see the different stages. They'll get a little bit bigger in these. These are a little bit in the early stages, and they'll start to develop on this long axis a primary root structure. And when you see that, it means you can transfer it to a different type of medium that begins to grow the plant as we're used to seeing it with the leaves and stuff. Especially in the 20th century, early 20th century, there were a lot of infestations of different types of fungus that basically decimated the cacao industry. One of the most recent ones, better said, was in 1995 in Brazil. It completely decimated the whole industry, especially in the provincia of Bahia. And from that, people started to think, we need to start to clone and produce different varieties that are more resistant to all the different sicknesses that are out there for cacao at the moment. But because of the self-compatibility, it wasn't possible, especially when you have a genotype that is really resistant, or at the same time producing a flavor of chocolate that is agreeable. There's fine aromatic chocolate, and then there's also bulk chocolate. Bulk chocolate comes from a genotype that's pretty resistant. The fine aromatic chocolates come from varieties that are more susceptible to the different fungal diseases like black pod rot, witch's broom, and frosty pod. Witch's broom actually attacks the leaves of young trees, so it kills the tree before it gets to produce pods. So after the embryos begin to develop and they elongate and they have a primary root structure, we transfer them, this is a good example, to a different kind of medium, and they continue to grow. The same material that comes from the previous plates, exactly.
So how old is this one here?
This one was from the same batch. This probably is about 10 weeks old at the moment. Okay. Could be a little bit older, could be a little bit younger. Once it starts coming with the secondary root structures, that's when we know that it's time to transfer it to media. Usually it's a perlite or a sand at the beginning, and we take it out of the sterile environment. This is called a magenta box. When we take it out of here, that's the time where it's most fragile. Cross our fingers that it'll survive, because until now it's always been in a closed environment, high moisture, and everything's been taken care of for it. So we do work a little bit with transgenic plants, cacao plants, meaning we find some DNA that reacts better to these pathogens, and we try to introduce it into the genome of a certain strain of cacao, and those are the transgenic, because it doesn't come from that specific plant. It's not as much as we would like. The pathogens are pretty fierce, and so it's been difficult. One of the most popular variety of bulk chocolate in the world is from this variety here, called CCN 51. It has a nice flavor, what we think about usually as what chocolate tastes like. Where the stem is connecting here, called a flower cushion. When we start the cell culture or the tissue culture, what we are cutting are these flowers that have not opened yet. Okay. And usually inside, they're still sterile. The fruits, which are basically a pod, a little bit smaller than a football. Each variety has a different size. But they grow directly from the trunk. They have lots of different colors depending on the genotypes. There's quite a large variety of. The reason why we have trees here, one is to take flowers for tissue culture. And the other is to have different varieties from around the world. One of the functions of our laboratory is not only to do our own research, but also to teach other researchers and give them a space where they can come and do research. What we have here is one of the centers of cacao research in the world. The cacao leaves, we can see a larger green one. Here's a mature leaf. And as they grow, they usually start out maybe as a lighter brown or copper color. And as they mature, they get darker and darker. If one tree is susceptible, it may wipe out your whole plantation. And that is one of the disadvantages of the clones. Oh, we have a great clone. It's very resistant. Disease comes through that it can attack that plant. You could take out your whole plantation and you'll have to restart. Those small little plants are taken out. We bring them to this enclosure. Told you before, this is a couple weeks of reckoning. Hopefully we're crossing our fingers that our plant will continue to grow. But you can see there's a meristem starting here at the top. So. And the perlite has a hydroponic system. We grow the cacao tree, and as you can see, it bends. And what comes out at the top, technically branches, that you can cut that and begin the propagation in the other ones. Thank you for coming to the Guiltinan lab. If you have any questions, we've got a web page and it's all up to date. So have a good day. One of the members of our lab, Drew Pfister, has developed a method for studying these diseases of cacao. Instead of using pods, which we have not been able to breed pods here because a lot of times we have the same genotypes that are not self-compatible, he developed a transient leaf assay. What it entitles is they grow up the fungus on a petri dish, and then from the petri dish they take a plug. This is the pathogen, exactly. From the petri dish a plug is taken and he places it on a cacao leaf that's a specific stage called a C4 stage. And we can see some of those in the greenhouse. And what he checks is how the different genes react to the fungus by using qPCR. So he's looking at the RNA production levels and things like that. He is able to see which genes are activated and which genes stay silenced, depending on the different types of pathogens that are introduced. And so this was a novel way to do this kind of testing basically in vitro, because with fungal spores, there's so many millions and billions of spores, we cannot enter and infect live plants in our greenhouses. So that was something that he developed in order to be able to study these diseases. So.